Hello, everyone. Welcome to the first press conference at the General Assembly of the European Geosciences Union. And this is our first of 8 press conferences this week. And there's a lot going on at the center. There are about 17,000 presentations, and we have around 15,500 participants. And 3 of them are here, giving our first press conference today. And we are live streaming all the press conferences remotely, so you can watch them online, and as well as a few other sessions that we have on. And we also have a number of press releases and other documents relating to press conferences, as well as other scientific sessions that are available in our press portal online. So if you go to media.egu.eu, that's our central press portal. And then if you add /documents to that, you will access our document repository. So people watching online that don't have the press handouts that we've distributed can get the documents through there. All right, so we can start this press conference. So I'll just quickly introduce the speakers, and then they will give their presentations. And at the end of all 3 presentations, we will open the floor for questions from the journalists here in the room and those watching remotely. Our first press conference is on health risks, air pollution, emerging viruses, and malaria mosquitoes. And the speakers are Patricia Tarin Carrasco. She's a PhD student at the Department of Physics at the University of Murcia in Spain. Elke Hertig, she's an associate professor at the Institute of Geography at the University of Augsburg in Germany. And Jan Willem Foppen, who is an associate professor of hydrology at AHE Delft, Institute for Water Education in the Netherlands. And I'll hand over to our speakers now. Thank you.
Good afternoon, everyone. I'm Patricia. My research is focused on the impact of air pollution on human health. Air pollution is a real problem on the society around the world. We have a problem, especially in big cities, where we can find more population. And with the climate change is expected an increase of the mortality and morbidity on the big cities. For this reason, the objective of the work is study the mortality cases due to fine particle matter through non-linear functions. First, we did an estimation through modeling of mortality cases for different age ranges for present climate, and we did a difference with future scenario. And we obtained results for Europe as 3 regions. On first place, we use this form for calculate the mortality and the risk ratio. And for the study, we divide Europe in 3 different regions according to the different characteristics. Is the East Europe, Center, and West. And for the simulation, we have we have 2 scenarios. The present one is from 1991 to 2010, and the future one is to 2031 to 2050. And we choose the RCP 8.5 scenario just to isolate the effect of climate change on the future mortality. And the mortality that we study is lung cancer, ischemic heart disease, chronic obstructive pulmonary disease, stroke, and the total is the sum of all the other mortalities that I say. We calculate for the different ages. We have different ranges. We have different mortalities, as I said, and we calculate for the different regions the mortalities. Okay. Here we can see the PM2.5 level concentrations. At the left, we have the present situation, and on the right we can see the different expected for the future scenario. At the present, we have that the hotspots are located in big European megacities and on the East and Central Europe. And for the future is expected an increase in south of Europe, and conversely is expected a decrease on the north of Europe. Regarding to the result of time of the research, on left, we present the total mortality. The unit is deaths per square kilometers. And on the right, we present the difference with the future scenario. We can see that the high number of mortalities are located in European megacities and on Central Europe. And for the future, we can see that is expected an increase of mortality number on the east of Europe. And here we present a specific mortality, in this case, lung cancer. At the top, we can see for the 65-69 range age. And at the bottom, we have the total lung cancer mortality. We have on the left of the screen the present situation, and on the right, the different expected on the future. We have the same pattern that the other case that I present, but we want to stand out that is expected a decrease of the mortality in the center of Europe, and on the other two regions is expecting an increase of the mortality cases. Just to conclude, having a detailed information about baseline mortality can help providing very detailed information about health issues. And the different type of mortalities caused by this pollutant, particulate matter, will increase in the future due to climate change penalty. And this climate penalty will affect mostly on the Western and Eastern Europe, increasing the mortality cases, while Central Europe will benefit with a lower concentration, decreasing mortality over these regions. And we should be in mind the aging of European population and decrease of city dwellers, variables that we don't have taken into account on this study, just to isolate the effect of climate change alone in the health of European citizens. Thank you so much for your attention, and you can follow our result of our work in our social networks. Thank you.
So I will be talking about the present and future distribution of Anopheles vectors, mosquitoes, and they transmit malaria, and I also assess the potential malaria transmission stability in Europe and the Mediterranean area. So I will only present some selected results. If you are interested further, there's a recent paper of mine. There you find further results and also the underlying data and methods. So why did I assess the Anopheles vector distribution and the malaria transmission stability? We saw a widespread elimination of malaria during the course of the 20th century in Europe and the Mediterranean. So before that, there were widespread occurrences of malaria cases, but due to land drainages, to wetland drainages, and to insecticide spraying and other measurements, malaria was decreasing in Europe and was eradicated. But recently, we see again an increasing number of malaria cases in Europe. And this is due to the international traveling. So people are coming with malaria from Africa, for instance, and bringing it again to Europe, and also because of the large number of immigrants also bringing malaria from the endemic countries. And so due to climate change and other factors, for instance, if the wealth of specific nations isn't that good as it is now in Europe, and ongoing urbanization, ongoing immigration, it's possible that we see a reoccurrence and or expansion of vector-borne diseases. And the mosquito occurrences are in general very highly related to climate. This means here you see the Anopheles mosquito life cycle, and here in the adult phase of the mosquito's life cycle, it's very sensitive to temperature conditions. It controls the active and the hibernation periods of the mosquito, so it needs a specific temperature threshold when it gets active, so temperature in spring, and in autumn when it again falls into the hibernation period plays a major role here. And we have also an aquatic life cycle of the mosquito, and here, of course, the mosquito needs water, and here precipitation governs the life cycle. I will show you results for one specific species of the Anopheles vector. It's Anopheles atropavus. It's the most widespread one in Europe. It's the dominant vector. For this area you see here in red, it's the occurrence probability during the historical period from 1985 to 2005. And it's not only the most widespread mosquito in Europe, it's also the one which has the highest proportion biting people. So there are different Anopheles species, and some of them only feed on animals, and other of them feed on humans. And Anopheles atropavus is the one in Europe which feeds for the most. It's only about 1/3 of the feeding is on humans, but it's very high for the European vectors. In comparison, the tropical Anopheles mosquitoes, they feed almost exclusively on humans. So that's the problem in Africa, that these mosquitoes have a different feeding behavior. And Anopheles atropavus has also a very high daily survival rate. It's a very resistant mosquito. So we see here that in the historical period, it's again the same picture here in the upper figure, what you've seen in the slide before. And here you see the modeled changes under climate change conditions in the middle until the mid of the century, and the lower one until the end of the century. And these are the changes from changes in temperature and precipitation. And what we can see here is that we have a northward spread of the occurrence, and this is mainly due to the temperature rise in spring and to precipitation increases in summer and autumn. So the projections show that we have here both temperature and precipitation increases, and this favors vector occurrences in Northern Europe. And what we also see, that there might be a decline of the occurrence in some Mediterranean areas. And this is mainly due to the precipitation decreases in summer and in autumn, which are projected for these regions. So this is the modeling of the vector distribution, where the mosquito is occurring. And I went a step further and assessed the vector stability index. And this vector stability index is used to assess the potential malaria transmission stability, which means it's not only the mosquito occurring, but also if it's possible that that malaria re-emerges in Europe and the Mediterranean area. And this vector stability index, it incorporates, for one, this human biting proportion, which are already addressed when I characterized the Anopheles atropavus. And also the daily survival rate goes into this vector, and the length of the extrinsic incubation period in days. And this is here a temperature-dependent variable. And you see here for the Plasmodium vivax, which is the main malaria parasite in Europe, it's you need a specific temperature threshold in order to be able to transmit malaria, in order to complete the malaria cycle. Currently, under present conditions, we have about 2 months where it's suitable in Europe, and when we project into the future, it will increase up to 4 to 5 months. And here are the results for the vector stability index. This is now not only based on Anopheles atropavus, but also on 5 other Anopheles species which are dominant in Europe. And we see an increase of the vector stability index, mostly here in the European, Southern European, Eastern European parts, increases where the climatic change favors the vector distribution, so that the mosquitoes occur, and also where the vectorial capacity is increased, which means that malaria transmission can occur. Both has to come together in order to increase this index. And here in the northern parts of Europe, we have more vectors because of the temperature and rainfall increases, but the temperature rise is not enough to impact on the malaria transmission. And here in southern Europe, in the southern Mediterranean, the vectors are missing. So the vectors are decreasing, so that's why you don't see here a high transmission stability. And if you look at this index, this has values until the end of the century under the high emission scenario RCP 8.5 up to about 15. Currently, in the endemic regions in East Asia and in South America, this index is about 5 to 8. But in the endemic regions in Africa, in the tropical Africa, this index ranges between 12 and up to over 20. So that you can use this number where to locate this transmission stability in the future. So there's also a poster session on Wednesday, and that's it. Thank you.
Good afternoon. I would like to tell you something which I think is alarming on the one hand and groundbreaking as well. The title of my presentation is Virus Communities with Adverse Human Health Risks Found in Groundwater Below Slums. My name is Jan-Willem Foppe. I'm a hydrogeologist, and I'm from IHE Delft in the Netherlands. I can do it like that. So this here is a simplified picture of the situation. On the left hand, you see a slum area where they usually have inadequate on-site sanitation, where they dispose of wastewater which finally infiltrates into the ground. Here you see groundwater. This here is groundwater, and there are particles in there. Here they depict virus particles. Downstream, this water is abstracted and used for washing, cooking, and bathing. And in the end, people can ingest virus particles. We sampled these groundwaters on a number of locations. No. Why is it not moving to the next slide? We sampled those waters, 31 samples we took, whereby we sampled for viral DNA, from various sources in 3 pilot slum areas, in 3 cities, in 3 countries in Africa. And the new findings are here in, I think it's blue, but it could be green as well. Green? Okay. We found viral DNA of 25 DNA virus families. Altogether, we talk about 200,000 DNA sequences in groundwater. 14 of these families are from above ground hosts. Then we can think of frogs, mice, rats, cows, horses, monkeys, and humans. Then we talk about 14,000 sequences. The human host viruses, the pathogenic ones for us in groundwater, were papilloma and various herpes and pox virus, about 4,000 sequences we talk about. And this papilloma is the one that is causing cancer. The one that we found is definitely causing cancer, not the one that we found there, but the type virus that we found is known to cause cancer. The implications of our research are, on the one hand, that we have limited knowledge of which pathogens can indeed reside in groundwater systems. That because of the location where we sampled in the groundwater system, we know that these particles have resided there for a very long time, so they must be very persistent. That people using this water, and it is used at a large scale, they are at risk because they ingest it. And we think that our research adds another new argument to the lack of good sanitation discussion that is continuing in the developing world, not this year, not the last few years, but for many decades right now. Let me go in detail. Let me try to do that. I don't know why this is. That is out there. So here we have the details, 3 pilot areas. First pilot area is in Greater Accra, a small village, 12,000 inhabitants. In Arusha, which is a fast-growing middle city of about 400,000 inhabitants. And in Kampala, in Uganda, the capital of Uganda, with about 1.5 million inhabitants. And 60% of these inhabitants, they live in informal settlements. Okay, so where did we take our samples? Surface waters, you see a photo of that here. Springs, this is an example of a spring in Kampala, where people fetch water in these yellow jerry cans. Duck wells, here we have a duck well in peri-urban Accra, where people fetch water in similar yellow jerry cans. And piezometers, where you can sample groundwater from various depths in groundwater. And we focused on depths ranging from 1 to 50 meters below the surface. 50. Finally, you end up with concentrates per sample. You see a photo of those concentrates there, which you then run through a sequencer. And we have more than 40 million DNA sequences that were of good quality that we could use for analysis, further analysis. So we found bacterial DNA, we found viral DNA, and we also found unknown DNA. Viral DNA, as I said before, 25 virus families, 14 families from above ground hosts, such as these frogs, mice, etc. New human pathogens, herpes, papilloma, and pox, here depicted in an artist's impression, based on science, of course. There was comparatively more above-ground viral DNA in groundwater in Kampala, and least in this peri-urban Accra groundwaters, which we think might have to do with population density, which is, of course, much higher in Kampala than in peri-urban Accra. Also, there was limited viral DNA material in surface water sources. There we found many rat pox and goat-related orf pox, it's called, which can be transmitted to humans. It works. The implications of our research, first of all, this groundwater system is apparently a virus storage reservoir. And some samples, they really took long travel times. We know that because of the position of the samples in groundwater system. So these virus particles, they have persisted there for a long time, and then we think about years. We appear to have extremely limited knowledge of which pathogens are capable of residing in groundwater. And 70% of all DNA we found was not recognized by any of the international databases that we accessed. So this is saved for future generations. Implications for people on the ground using this water. These viruses, herpes, pox, and papilloma, they can result in skin-related diseases. The papilloma virus type we found can give rise to various types of cancer. In one spring in Arusha, we found DNA which is very much related to monkeypox virus, which has a mortality of 1 to 10%. And that monkeypox virus is a kind of a special, rare type of virus. But we're not finished yet, because we don't know whether the virus particles that we found are alive, or are active, or have still a certain infectivity. We don't know whether there are still other viruses and harmful bacteria out there. What we sampled is DNA viruses, and there's another huge group of viruses, which is RNA virus. We did not have a look at that. And of course, one of the most important questions is how can we improve then sanitation on the one hand and sources of drinking water on the other, or ideally both. And then you have to perhaps think about, let's start doing something about sanitation and improve sources of drinking water. In the project that we do, which is called T-Group, we experiment with an approach which is called transition management, which is more of social science, whereby we do some co-creation of knowledge with local communities and authorities to collectively define and identify pathways towards a healthier environment. This is the group, and here we have the funders. Thank you.
Thank you very much for your presentations, and my apologies for the technical issues we've experienced.
Accepted.
Thank you. We can now open the floor for questions.
Say your name and affiliation. I'm Jochen Steidler from the Austrian Press Agency. I have a question to Patricia Tarin-Carrasco. You mentioned that the fine particle matter concentrations in Central Europe, like in Germany and Austria, are declining. Is this just because of the climate change, or is it because of political actions?
No, it's for the climate is we call climate penalty.
Yes.
Yeah, for the will decrease for some I don't know technically to explain. For the temperatures and the different reactions, PM 2.5 will decrease in these areas.
Okay, so because of change in the weather, change of precipitation, change
yeah,
and stuff like
will
that.
increase the precipitation on north and part of the center of Europe. Is conversely than on the south. We expect on the south less precipitations. Then the particulate matter will stay on the atmosphere and is for that.
Okay, perfect. Thanks a lot.
All right, any more questions? Just say your name and affiliation.
Hi, it's Adam Vaughan here from New Scientist. LKO, just wanted to ask, obviously no one wants more mosquitoes, generally speaking, in Europe, but it would have sort of, looking at your maps, a slightly flippant way of looking at it seemed to be that actually we don't have to worry because in the Nordics, it won't be high enough to have malaria. And in the south, actually, it's going to start to get so dry that the vector, the mosquitoes just should we really be worried is what I'm asking from.
So the vector stability index increases particularly in the southeastern European parts, parts of Spain, parts of southern France, Italy, and lots of the Balkan countries, Greece also. So yes, I think there should be awareness. Currently, I don't think that it's possible to have really a widespread re-emergence of malaria because our health systems are quite good. But it's a long time until the end of the 21st century, so we should be aware that it might be a cure. From a climate point of view, it might happen.
Are there any additional questions, either from the journalists here in the room or those watching remotely?
Milan Ilic, newspaper from Ljubljana. I live here and I report for them. One question for Mrs. Carrasco. Did you factors that how to say have influence on the lung cancer and COPD, like exposure to asbestos or smoking?
No, for you can finish it. Okay. For this work, we only take into account how is expected the mortalities just with the action of climate change. We don't take into account the movement of city dwellers on big cities or the agents of dwellers in Europe. We take only particulate matter and the action of climate change.
Okay, and of course, question for each one, Professor Kartik. Okay, malaria is maybe potential problem, but there is already a problem. I was 10 years ago, the medical doctors from Germany said that a few decades ago they did not have specialists for meningoencephalitis caused by insects. I don't know what is the English name, and it is spreading with the climate change. Actually, probably the climate change. It is spreading now from Austria and into Germany. So, do you have also data, or are you doing, or your colleagues on that, the connection between climate change and the spread of from south to north of fusarium meningoencephalitis or meningoencephalitis?
It's planned now in the near future to also assess this. Yeah, we are not only interested in Anopheles and malaria, but also other vector-borne diseases, and this will be one of those we want to look at, but we haven't done it yet.
And finally, one practical question for Professor Poppen, because we saw that your sponsors and supporters are not only a purely scientific institution, but also socially, somehow their intention is social improvement. Actually, in local communities and on the state level, even in Africa, after, I don't know, 60 years after the end of the colonialization, etc., are they aware that one of the basic function of the state on all levels is to provide the basic infrastructure? And the basic infrastructure is like, okay, an example in Europe is that there are protected regions for water sources. I mean, that is the basic things. If you have a mayor, even on a local level, in a village or a small community, to have the secure water. I know that it is in practice is impossible with informal dwellings, but to have like national, those countries have national parks, have regions that are locked, protected as water sources. That everything else is counterproductive.
You are touching a very complicated issue, which we as scientists would like to call a complex problem or a persistent problem, for which there are no simple solutions. I think then we get into more of a discussion than that. I think local authorities are, and that depends on the countries where we work in, but they are very much aware. Especially when we talk about Kampala, the local authorities are extremely aware of what is going on there. Our finding will actually assist them in trying to implement their policies. And their policy is actually to close down as much as possible the polluted springs. Because the poor living in those informal areas, they use these free springs. But if you close down the springs, then you take the only source of free water away. And you have to remember that 60% of the population of Kampala lives in these settlements, which is a huge voting body. They have influence just because of the sheer numbers. So it's very complex political issue. You cannot, as National Water, which is the authority there, National Water and Sanitation Corporation, you cannot just close down the springs. There are no secure springs. That is the problem.
Thank you very much. Are there any additional questions? If not, we can finish here. You are welcome to approach our speakers. Now that we've finished, there are interview rooms available that you can book outside at the info desk. And we are done here at the Press Center for the day, but there is another press conference tomorrow starting at 9 on the quest for 1.5 million year old ice. So I hope you join along to that. And I'll thank again to our speakers and apologies again. Thank you very much.